food or a mate or like a new habitat.
Yep, correct.
And then the different types of movement, you have the cellular level, the WBC leukocytes. It has an amoeboid movement. That they move through pseudopodia-like projection.
Yes, correct.
And then muscles, it has muscular movement, which is usually contraction and relaxation of the muscles.
Right. So this comes under the category of contraction and relaxation movements, or also known as the muscular movements, where basically the thing which is moving is it stays at the same place, but it makes the whole organism locomote. So through muscular movement, the locomotion of an organism happens, correct?
Sir.
Yeah. Yeah, continue. And what is required for a locomotion to happen? What are the major components? Only muscular movement?
You have skeletal and then neural coordination.
As well. Yeah, correct. Yeah.
And then you have ciliary movement. So most of our internal tubular organs are lined up by the ciliated epithelium. Coordinated movements of the cilia, that is in the trachea, it helps in the removal of dust particles, and then in the fallopian tube, in the directed motion of ovum.
Correct. That's ciliary. Right. I also told you there is another kind of movement that can happen at a cellular level, which happens in human sperm and euglena, called flagellar movement. Okay.
Yes, sir.
Yeah, this is something just you should know for your entrances. Okay. Yeah. Then.
Should I move on to classification?
Yes. Just the muscle. Then we started with muscle, right? We started talking about, because the idea is that we have to study locomotion and movement in humans. It is under the human physiology part. So I just gave you some common outline, fundamentals of how things work on a cellular level. Now we are coming to muscles. So let's first talk about what are the characteristic properties of muscles, the major characters that it should always have.
Okay, so in skeletal muscles, it's voluntary.
No, before that, the characteristic property of every muscle, like in general, what muscle should be like. You can see the screen, right, Jania? Yeah.
So excitability, contractibility, extensibility, and elasticity.
Yes. You can remember it as E3C. Excitability, which means muscles should be able to be, you can excite the muscles. Okay. They should have contractility. They should be able to contract and relax under different conditions. Extensibility means once extended, they should have the load bearing capacity. Like you can extend it to its limit and it should hold there in place. And elasticity is just like a rubber band. It should be elastic, and once extended, it should come back to its normal shape. So these are the four characteristic properties of muscles in general. Okay.
Yes, sir.
Yeah.
Okay, so then for classification of muscles, you have skeletal muscles, visceral muscles, and then cardiac muscles. Skeletal muscles are voluntary muscles, and then visceral muscles and cardiac muscles are involuntary.
Yeah. So you will have, questions on this part, as write down differences between different types of muscles in the body. So first thing you will write is there are three types: skeletal, visceral, and cardiac. Where are skeletal found?
Bones.
Yeah, so write down skeletal muscles are found attached to bones or the skeletal system of the body. Skeletal muscles are found attached to bones or the skeletal system of the body. Okay.
Yes, sir.
Then where are visceral muscles found?
In the inner walls of hollow structures in our body.
Yes, they are found located in the inner walls of all our visceral organs, like stomach, which is alimentary canal, reproductive tract, urinary tract, etc. And where are cardiac muscles found? This is the second point. Where are they found?
Heart.
Heart. Okay, they are the muscles of the heart. And third point you will write is that skeletal muscles are striated and visceral muscles are smooth. But cardiac muscles are involuntary and striated. Correct? Skeletal muscles are voluntary and striated. Visceral muscles are involuntary and smooth. And cardiac is a mixture of both. Okay.
Yes, sir.
So they are striated but involuntary. So we have been joined by Devangi. Good evening, Devangi. How are you?
Good evening, sir.
Doing good. How are you?
Yes, sir, fine.
Okay. How is it at your place, Devangi? Where are you? Is it cold? And at your place, Jania?
Yeah,
Yes, sir.
actually, winter just came right now for us.
It just came right now. Where are you? Where are you situated, Jania?
Abu Dhabi. So even December it was
Ah.
actually kind of hot. Like towards the end, it got cold.
Oh, is it? That's good. And I think Devangi is in India.
Yes, sir, very cold.
Yeah, where are you, Devangi? Delhi only?
Ghaziabad, Uttar Pradesh.
Ghaziabad. Yeah, it must be. Yeah, the north is very cold. Because I am in Delhi NCR, so it's very, very cold. And it has been cold from like some time now. But so if winter has also come at your place, Jania, take care, okay? Because your boards are your exams will be coming. When are your final exams, by the way? We also have to keep a track. Anything declared?
Our principal, like during assembly, he said it'll be around like maybe like February, maybe third week. Yeah, like around the middle. Yeah.
Yeah, like mid or end February, third week. And what about you, Devangi?
Same, sir, mid-Feb.
Mid-Feb. Okay, cool. Great. So let's move with locomotion. Okay, so that's it. And today, let me tell you about some muscle. So how is muscle? First, we will talk about skeletal muscles. So how is skeletal muscle? Write down the architecture of skeletal muscle or the structural details of skeletal muscle. Yes. Now, as we know that skeletal muscles have to be bearing the load, they have to help in movement and all, correct? So let me take this picture from NCRT that I've taken. Yeah, can you see this image, all of you?
Yes, sir.
Yeah.
Yes,
This is a
sir.
picture from your NCRT. And let me make it big. Is it clearly visible now? Okay, very good. We can make it a little big. Yep. So look at here. So this what you see as whole. So this orange layer, if it is visible, this whole is a muscle fiber. Okay. So it's one big fiber, one muscle. The whole. So each this thing here, which you are seeing here as like beehive structures, which I'm drawing with blue, these are long fibers that are running inside like this. Okay. This is a cross section. So it's like you cut and you see these long. So these cells are like this. So this is how these cells look like. They have a okay, striated, and they will be having nucleus and also branching. So these fibers will run long inside, and together they will form a bundle. So each muscle fiber is a muscle cell, also known as myocyte. Myo means muscle, cyte means cell. And many such myocytes will together form a fascicle, which is a muscle bundle. So this one, let me use green color. It's slightly different. So this one bundle that you see here is your one fascicle. This another bundle is another fascicle. It's like, so individual threads or these individual fibers are like threads. When they come together, they make a bunch called a bundle. All these bundles come together to make a big muscle chunk. Okay. Is it clear? And so is it clear, everyone? So first write down a fascicle. A fascicle is organized skeletal muscle. This name comes from a word called fascia. Okay. So write down each fascicle is an organized skeletal muscle bundle which is held together by common connective tissue layer. Common connective tissue layer called fascia. And fascia is made up of collagen. Common connective tissue layer called fascia. And fascia is made up of collagen. So it's collagenous tissue layer, collagenous connective tissue. So it's like, have you seen wirings? So many wires are put together and they are bundled by using tapes, right? In electricity.
Yes, sir.
Yeah, that's how our muscles are also organized. So many muscle fibers, individual fibers come together and they are held together by a covering, a layering, which is called fascia. And fascia is made up of collagen. So it's like a collagen tape, which is over these fibers, and it makes a muscle bundle. And many such bundles then come together to make a whole muscle chunk. Okay. Now, next thing, write down. So these muscle fibers are also associated with blood vessels. So you can see blood capillaries, right? Sarcolemma. Anyone knows what is sarcolemma? Sarcolemma. Sarcolemma is what covers the muscle fibers. Okay. So the word sarcolemma comes from the word sarco, sarcox or sarx. It is a Greek word which means flesh or sheath. So if any one of you is a non-vegetarian, then what I'm saying is because you must have experienced a fleshy sheath around muscles while having a chicken. So that is sarcolemma, and it's a proteinaceous sheath. Okay, it's made up of protein, and it covers the fibers. So write down sarcolemma is a transparent sheath. It's a transparent sheath that covers the muscle fibers, and it is present only in skeletal muscles. Covers the muscle fibers of skeletal muscles. Is it clear? Thumbs up. Very good. Now coming to blood capillaries, so write down muscles are associated with blood vessels, very fine blood capillaries that provide the muscles with nutrition and oxygen. That provides the muscle with nutrition and oxygen. Now the interesting thing is that this sarcolemma is it acts like a plasma membrane, okay, because it's lining the muscle fiber, each and every muscle fiber. And in this sarcolemma there is a fluid filled. So this covering has a fluid which is called sarcoplasm. So write down next point. So sarcolemma is like the plasma membrane for the muscle fibers. Plasma membranes of muscle fibers containing or enclosing the sarcoplasm. Just like cytoplasm, which is inside the cell, nucleoplasm, which is inside the nucleus, because this is covered by sarcolemma, it is called sarcoplasm. Is it clear, everyone? Thumbs up if it's clear. And this sarcoplasm is multinucleated. What does this mean? What does this mean? It means it has multiple nucleus in it, which makes muscle fiber a syncytium. So as I told you, muscle fibers don't have one single nucleus per cell because they are long fibers. So it is difficult to define one cell where one ends and the other starts. So their sarcoplasm contains multiple nucleus, and you can understand why, because nucleus is very important along with mitochondria for regulation of a lot of activities, especially energy production related things, right? And for gene expression. So more protein formation, more genes have to be expressed, more wear and tear. So muscles have to be muscles have to express a lot of genes. So that's why it's called a syncytium. Syn means where it's connected, it's joined. So it looks like muscle fibers are all joined to each other with multiple nucleus being shared. So sarcoplasm is multinucleated, and write down that's why muscle fiber is called a syncytium. Is it clear? I think this is the correct spelling. Okay. Now, in this sarcoplasm, a characteristic feature is that there are small filaments. Just like these fibers are called myocytes, these there are filaments present in the muscle which are called myofilaments or myofibrils. So let's also write what is myofibrils. These are just words that you should know. Myofibrils. The word itself says the fibers of muscles or filaments of muscles. Write down. Myofibrils are muscle filaments. These are muscle filaments which are arranged parallelly. Which are arranged parallelly in the sarcoplasm. Okay. Arranged parallelly in the sarcoplasm. Now let's go into some little bit detail and understand what is these filaments made up of. So these filaments, which are arranged parallelly in a sarcoplasm, they basically, remember I told you that striated muscles are called striated because they have light and dark regions. Yeah.
Yes.
Yeah. That light and dark region actually comes from, these are actually called light and dark bands. And it is because of two important proteins present in the muscle, which is called actin filaments and myosin filaments. Okay. So these filaments, which are arranged parallelly in our sarcoplasm, first write down these myofibrils or these myofilaments have dark and light bands on them. These microfilaments or myofilaments have light and dark bands on them due to the presence of actin filaments and myosin filaments. These are two proteins present in muscles which makes our muscles move. Actin and myosin. Is it clear, everyone? Okay. So this actin, wherever actin is present, it is called the light band. It gives a light color appearance. Okay. And wherever myosin is present is called the dark band. So light bands are due to actin and dark bands are due to myosin. This part is clear? Simply? Very good. Now let me also pull out another figure which will explain this thing better. Yep. This is again from your NCRT. Can you see this figure? All of you? Both of you can see this? Yeah. So now pay attention to what color should I use? Let me use blue. Okay, now pay attention to this figure and look here. Do you see that from here to here there is a dark band, then there is a dark band, and then there's a dark band, right? There's a small line which is dark here, a small line which is dark here. And we talk about the light bands. These are the two light bands, and these are the light bands, right? Now each, if you pay attention, they are like fibers. They are individual fibers. They are like this, correct? Here also, they are like these fibers, right? So wherever, first you have to understand these, keep writing certain things. So wherever you find a light band, okay? So from here to here, okay? And here to here. So together, we take it from here, okay? Let me take it, this region. Okay, it's not, okay, should go, yeah. So this region is called the I band. What it is called? The I band. In the I band, there is only actin present. So this is actin and this is actin. These are two different actins joined together by this line. So this line which joins here, this line is called the Z line. Okay. On the both sides of Z line, there are actin attached. So if the question comes in your entrance is, actin is attached to which line? I, A, Z, H. Where is actin? So actin is present in the I band. This is called the I band. In the middle of I band is a line called Z line. Okay. So you can see that this is an actin, correct? And this is an actin. So it is attached in an alternative manner going to either sides. Make sense? Say yes or no. I band and Z line is clear? Perfect. Now write down, wherever there is a dark band, it is called A band. So this is A band here. And in the A band from here to here, this is all myosin. Okay. So let me use green. Okay. And this is one myosin. One myosin interacts with two actins here and here. So it's in between. Can you see this, everyone? Is it clear? Now they have also defined another zone. Let me use a light color and show it to you. So can you see that the zone between this and this is actually a dark band, but it is slightly lighter. So the shade from here to here and here to here is same, but this is slightly lighter, correct? That is where only myosin is present. This is called H zone. So one question that has come is, what is H zone? So in the H zone, only myosin is present. Write down, in the H zone, only myosin is present. Whereas in the A band, both myosin and actin is present, because A band is from here to here. So this is more denser because there is actin also, there is myosin also, then actin also, myosin also, actin also, myosin also. So this region of the A band, okay, is more denser. Here it's just myosin. Here again actin, myosin. here it's just actin. So write down in the I band, it is just actin. In the A band, it is both actin and myosin. And in the H zone, which is present in the center of the A band only. So H zone is not something different. It is present in the A zone only, in the center. There is only myosin present. And if I tell you what is a sarcomere, so between one Z line to another Z line is one sarcomere. Is it clear? That is one sarcomere, which is making our muscle fiber. This part is clear, everyone? Very good. So you have to read this part from NCERT where the same thing is written and just read it in detail. Okay, know about all the zones, in which zone. And can you see that it gives a black and white, black appearance? So this is what gives skeletal muscles their characteristic striated appearance if you look it from outside. When you look it under the microscope, it is because of the density of actin and myosin. Where both are present together, it's darker, or where only myosin is present, there also it's comparatively darker. So this zone is darker than this zone. This is the darkest because here both are present together. So myosin is in A band only and it is darker, whereas actin is an I band and it is lighter, and H zone is slightly lighter than the dark zone and darker than the I zone, where only myosin is present, no actin overlap. Okay. Now remember this H zone, it is very important. And why it is very important, I'll tell you. So this part is clear. You want to note down, draw, please do it and let me know when it's done. Please write in the chat when you're done, people. I'll just take a minute's break and I'll be back, okay? Is it okay, Devangi and Jania? Please note these things down. Yeah. Okay, you can see that Devangi is done. What about you, Jania? Shall we move forward? Okay, so if this is clear to you and all the bands, colors, codings are clear to you, now let us look at the structure of these two proteins. So write down structure of actin and myosin. As I told you, they are like filament proteins. Okay, they are actin filament and myosin filament. Actin, myosin. They are also known as contractile proteins because they give the muscle the ability to contract and relax. Now, how does it happen? We will talk about it. So actin and myosin are filamentous proteins or contractile proteins. Okay. Now there are two type of actins. Okay. One is called F-actin. Okay. F-actin. Basically, it's the protein is same, but one is F-actin and one is G-actin. This is not in your NCERT, but just know F means filamentous. It is like a rope, long chain. G is globular. These are monomers, single units. So G-actin is this. It's like one round. But F-actin will be like a filament. Make sense? So if these units are single, they are just called G-actin. But if they come together like the chains, links of a chain and make a long chain, they are called F-actin. So we have to focus on this part. Okay, not this. So this is what is present in muscles, F-actin, known as filamentous actin. So write down first each actin filament is made up of two strands of F-actin. Two strands of F-actin which are helically wound to each other or which are present in a helical manner. So two F-actin filaments will be there, which will be helically, just like a DNA helix, they will be helically bound to each other. And two another filament, write down two another filaments of tropomyosin. So this is a different myosin. It's called tropomyosin. Is associated with F-actins. So let me pull out this figure for you. Okay, just 1 second. Just give me one moment, people. Pulling out that figure. Okay, you can come to figure 20.3 of your NCRT, everyone. Yeah. Can you all see the screen still that is being shared? Devangi, Janya?
Yes, sir.
Yeah. So just one moment. I hope, yeah. Can you see this figure that I have pulled in?
Yes, sir.
Yeah. Oh, but the screen is malfunctioning for me. Is it also malfunctioning for you?
Yes, sir.
Yeah. Okay, anyways, let's try to make this big. Hopefully it will solve. No. Come on. Yep. Okay. I hope you can see this, right? So we can spend some time on this. Is it clear? Everyone can see this? The tropo. Yeah, okay. So this, what you see here is actin, actually F-actin, because it's filamentous. Can you see this each? If I use this one, so this each ball here, which I'm highlighting, is a G-actin. Okay, is it clear? So this is a G-actin. When this G-actin comes together and make a filament, this becomes a F-actin. So can you see there are two F-actins, one strand and second strand, right? And they are wound together in a helical fashion. And on top of it, this rope-like structure that you see is tropomyosin. So tropomyosin keeps F-actin in place, and together this is the whole actin filament. There is some small proteins which are present at places. So this is called troponin. So what does troponin do? It binds tropomyosin to F-actin. Is it clear? So write down. Oh, sorry. It doesn't bind. Oh no, this is not working. Let me restart this thing, people. Okay, in the meanwhile, can you all see the figure in your NCRT?
Yes, sir.
Yeah. So just write down the protein troponin. Till I'm opening it, you can write down the protein troponin is present at regular intervals on the tropomyosin protein. The protein troponin is present or is distributed at regular intervals on the tropomyosin protein. Yeah, can you all see the screen back? So this troponin actually sits exactly at the place which was the binding site for myosin. So what you see here is actin filament, and this is a myosin filament. Now, can you see these head regions which says these active actin binding sites? Basically, these two go and bind here. I'm making it ulta. So these sites go and bind here. But because troponin is sitting there, it acts as a masking agent. Okay. Is it clear? Make sense? Yeah. So till troponin is there, myosin won't bind. So you can write down the troponin in the resting state, the troponin in the resting state, when your muscles are resting, masks the active binding site for myosin, or covers, you can also write cover, or covers the or hides the binding site for myosin. So this is about the actin. This part is clear. Tropomyosin, effectin, everything is clear about this molecule, this filament. Now let's come to myosin first. Myosin before going further. So a myosin, you can see that there are two monomers. Okay, so two things intertwined together like this, and it's like a two-headed snake. Can you see it? Myosin looks like it's a thick filament, and write down, it is a polymer protein. It's a polymer protein. As you can see, there are two here which comes together and are present again like in a wound manner, like a double-headed snake. And in the head region, there are two sites, as you can see. Let me make these things a little clearer. So one site I will mark with red, which is this one. Can you see these two sites? This is called ATP binding site. So for contraction and relaxation of muscles, we need ATP, right? Energy.
Yes, sir.
So those ATP molecules bind here. And when they bind here, now this myosin with its cross arm in the head region, let me use yellow color, goes and binds to actin with these regions. So with yellow, I am showing you the actin binding region. So these are the actin binding sites. Both things clear? So in the head of the myosin polymer, there are two sites, ATP binding site, and in each head there will be one ATP binding site and one actin binding site. So there are two heads. So there are two actin binding sites and two ATP binding sites. So this whole story cut short is clear to you, right? We are going step by step. So this has to bind here, but troponin is present, so it cannot bind in a resting state. Okay. Please note and write down, and write in the chat when you're done. I will encourage if you draw this diagram by your hand and write and label every component yourself. Take 2-3 minutes, not a problem at all. Draw it and write in the chat when you're done. Is it done, people? Please write in the chat when it's done. Okay, for Jania, it's done. What about you, Devangi? Yes, Devangi has a very good question. So Devangi, can you just hold that question for some time? When the muscle is relaxed, so Devangi's question is, when the muscle is relaxed, myosin will not bind to actin. So will it affect the striated appearance? It will change the appearance a little when the muscle is in working, contraction versus relaxation. But the striated appearance will still be there because though it is not bound, it is present. Correct? It is just not bound, but these two, like here, can you see? So this is myosin, this red one, it is still there, and this is actin, it is also still there. They together are not bound here. If I use, so this binding has not happened, they are unbound, but they are present in the muscle. Make sense? So the banding is because of the presence. Is it clear, Devangi?
Yes, sir.
Now this banding pattern will slightly change in a contracted and a relaxed muscle. That's true, but still there will be banding. So light and dark bands will always be there. Does that make sense, Devangi? It's a beautiful question.
Yes, sir.
Yeah. So exactly that I'm going to tell now. So if this part is clear to you, let's move to the mechanism of muscle contraction. Okay, this is the most important part of this chapter. For this, we were building this whole thing, fundamental concepts and all. So this is the mechanism of muscle. How does our muscle contracts? Through these two proteins. And this mechanism is very famously known as the sliding filament theory. Sliding filament theory. Okay. Now, this word must have given you some idea in itself. So there are two filaments we know, actin and myosin, and they are sliding over each other. That's how muscle contracts and relaxes. How many of you have seen that old slider phone that used to come? Any one of you has seen a slider phone? The top part, the screen, used to slide over the bottom part where the keypads were there. So these are the keypad one, the phones one. Do you understand what I'm talking about, people? So there used to be like this. So you will just see a screen with one button. But from side, if you will see, there will be two units attached to each other. And this is what you are seeing from the screen. So when you will press this button and put a force up, the screen will go up with the button. And from below, there will be a keypad that will come out. That's called slider. You understand?
Yes, sir.
Similarly, there was a foldable phone also. This was the screen and this is the keypad, and it will fold on itself. And in other case, it will slide. So similarly, it's a sliding theory, sliding filament theory that justifies and explains how muscles work. So what happens first? Let's write a certain things. Write down. So now you know what are thin filaments and what are thick filaments. So actin are thin filaments. Write down if you have not understood. Actin are thin filaments and myosin are thick filaments. Okay. And according to this sliding filament theory, the thin filament slides over the thick filaments. And that's how the muscle contracts. So write down. The sliding filament theory states that contraction of muscles like which are the fibers, myofibrils, takes place by the thin filaments which are actin filaments sliding over thick filaments which are myosin. Okay. Is that clear? Now, if this sliding is happening, there must be a signal for this function to happen in the body, right? So how does muscle contraction begins? How does the body knows that? How does like your leg muscles know before you decide to wake before you decide to stand and start walking? How your muscles know that, okay, now I have to start contracting and relaxing to initiate locomotion? Who controls? Because it's voluntary. So we say that we can control, but how can we control it? Through which organ of the body? We can control it through the brain, right? The central nervous system. Yes or no?
Yes, sir.
Yeah. So the signal for the contraction of muscle, write down the signal for the contraction of muscle. The signal for the contraction of muscle comes from the central nervous system. Comes from the central nervous system through a motor neuron. Through a motor neuron. When we studied neural control and coordination, I told you about sensory neurons and motor neurons, correct? Remember that? Devangi, Jania, and Aisha? Yeah. So the signal comes through motor neurons. And I told you that there is a neuromuscular junction between the muscle, which means the sarcolemma, and the neuron. So that is called a neuromuscular junction. This is where that neuron comes and gives a signal. Okay. So write down next point. This motor neuron forms a neuromuscular junction. Forms a neuromuscular junction with the sarcolemma. The sarcolemma which you just studied above. With the sarcolemma and release neurotransmitters. Remember neurotransmitters? Chemical synapse. Devangi, Jania, Aisha. Yeah. So they secrete neurotransmitters. In this case, just remember the name of the neurotransmitter. Sometimes it's asked in the entrance. So at the neuromuscular junction, the most abundant and the most common neurotransmitter is acetylcholine. Acetylcholine. Okay. So whenever there is a secrete of neurotransmitters, what happens? A signal is sent from neurons to the next cell. I told you in some cases, in most of the cases, that neuron sends a signal to another neuron, but it can also send a signal to muscles. So if it is a neuron making a junction with another neuron, what will that be called? Quickly. Jania, if a neuron is making a connection with another neuron, what will that be called? One word. Devangi? Yeah, Devangi is writing in the chat, I guess. Yes. Oh, Jania is writing. Great, Jania. It's called synapse. It's called a neural synapse. But if a neuron is sending a signal to a muscle, it's called a neuromuscular junction or a neuromuscular synapse. Okay, so a neural synapse and a neuromuscular synapse. In that synapse, always neurotransmitters are released if it is a chemical synapse. In this case, the neurotransmitter is acetylcholine. Okay. And this will generate, write down next point, This neurotransmitter release, keep writing it as points. This neurotransmitter release will generate action potential in the sarcolemma. Again, I hope you remember action potentials. That's why we first did the neural chapter. Remember action potentials, people? The signals which are generated within the neuron, in the membrane. Yes. So action potential is generated, which spreads throughout the muscle fiber. Which spreads throughout the muscle fiber. Okay. So let's see what has happened till now. Till now, there is a signal sent by the brain, CNS, via a motor neuron. That motor neuron has come and made a junction with the sarcolemma of the muscle, and it is called a neuromuscular junction. In the synapse of that neuromuscular junction, the neurons, the motor neurons, will release neurotransmitters, chemicals. In this case, it will be acetylcholine. This release of chemical will generate action potential, which will spread throughout the membrane of the sarcolemma. Okay, throughout the muscle. Clear till here? Thumbs up. Great. Now, after that, what will happen? Due to this action potential, calcium ions will enter into the sarcoplasm. Write down. Calcium ions influx. Influx means going in the sarcoplasm. Calcium ions will move into the sarcoplasm. And now is the story that will make sense to you. This calcium binds to troponin. Okay, and remember what was troponin doing? Devangi asked that question. Troponin was covering the binding site. So till you were resting, till there was no signal, there was no calcium in the sarcoplasm. And because there was no calcium, that calcium was not going and binding to troponin. And because it was not binding to troponin, it could not remove troponin from where it is sitting on the tropomyosin. So the binding site for myosin will be covered. When calcium will go in, it will bind to troponin and will remove the troponin from the active site of myosin. And now it is free, right? Story is clear till here? Very good. So write down, calcium binds to troponin and remove it from the active site of myosin on the actin filaments. Remove it from the active sites of myosin on the actin filaments. Okay. Now, because this active site is now free, so myosin can bind, right? Myosin can bind. But for everything to happen, any binding, unbinding, because myosin is also a protein, actin is also a protein. You need energy, right? Yes or no?
Yes, sir.
Now, that energy will come from ATP. So write down. Now, myosin head will utilize the energy from ATP because ATP can come and bind to the ATP binding region in the head. Remember, there were 2 binding sites. That's why I showed you that. So ATP will come and bind to the ATP binding site. And this myosin will break the ATP into ADP and P. And that breaking of bond will release energy. That energy will be absorbed by myosin, and it will use that energy to bind to actin. Okay. Is it clear? So what we have achieved till now, let's say if in black, let me erase it. Let me use. So let's say if in blue, I am showing actin. Okay, this is actin. And in red, oh, so this is one head. This is another head. This is myosin. So now what I have shown is that this can go and bind to the active pocket. And this binding, it looks like a bridge, right? Makes sense? The binding has happened. Is it clear, everyone? So write down myosin head binding to the actin forms a cross bridge. Okay.